Okay, many thanks, Michaeline, and I want to thank Life Technologies for giving me the opportunity to present today the results of this collaborative effort of 8 European laboratories which collaborated to build up this panel for hotspot mutation screening in colon and lung cancer. Now, before getting to the results of what we did to develop the panel, I would like to make a brief introduction in order to provide the basis, the need for this type of tools in clinical research, in particular in colon and lung cancer. Well, as you might know, the identifications of the mechanisms that are involved in the proliferation survival of cancer cells has allowed in the past 2 decades to develop a number of new drugs that we refer to as target-based agents, which are able to specifically block molecular mechanisms that are involved in tumor pathogenesis. And there was a great expectancy from the activity of these new compounds for the treatment of cancer individuals with cancer. However, we have also to acknowledge that most of the clinical trials that have been run in the past decade failed to demonstrate any significant activity of these drugs. And this basically happened because these trials enrolled unselected populations of subjects with different tumor types. Why do we need to select individuals? Why do we need to select tumors? While tumors are extremely heterogeneous at the molecular level, and we have to recognize 2 levels of tumor heterogeneity. The first is the intertumor heterogeneity. That means that if you take the tumors originating from the same organ but from different individuals, in most cases you will find that these tumors have a different molecular profile, and this molecular profile can be significantly different. In addition, within each tumor, we can now recognize the presence of different clones of tumor cells which carry distinct molecular alteration. And as we will see, both these intertumor and intratumor heterogeneity are really the cause of the resistance to target-based agents in the treatment of cancer. Now this has been finally recognized, and the development of any new drug in cancer is always associated with huge biomarker discovery program in order to identify those molecular alterations that might predict the sensitivity or the resistance to the drug, and therefore that allow to select those tumors that can really respond to treatment. And a further development of this concept has been called as genomics-driven oncology, something that is being discussed in many meetings this year, starting with the ASUR meeting. The idea is that now we have available new tools that can allow us to perform a comprehensive molecular characterization of the tumor coming from any individual, and so having this information we can choose the most active compounds and try to block the tumor using such new drugs. In this respect, I have to say that in both lung and colon adenocarcinoma, it has been already possible to identify a number of subgroups of tumors which carry specific molecular alteration that might offer potential for therapeutic intervention using new drugs. However, there are some limits to this approach that we must acknowledge. The first is that the fact that a tumor has a mutation in an oncogenic pathway does not mean that that tumor will respond to the corresponding inhibitor. Why? Because tumors carry dozens of different molecular alterations, and only in few cases it has been possible to demonstrate that the tumor depends from a single pathway, and therefore by blocking this pathway it's possible to significantly inhibit tumor growth. This phenomenon has been called oncogene addiction and needs to be demonstrated at the preclinical and clinical level, and here in this box, in this black box, we will see a partial list of the few subtypes of tumors in which this phenomenon has been formally demonstrated to occur. Another important point is that the majority of tumors that initially respond to target-based agents then become resistant at a certain point. Now, emerging evidence suggests that the seed of the resistance is present in the tumor before we start the treatment with the target-based agents. In particular, what is emerging is that the tumors are extremely heterogeneous and that subpopulations of tumor cells that carry resistance mechanisms are present in the tumor before treatment, and therefore treatment is simply selecting the resistant cells that will grow up and will be responsible of the recurrence of the disease. Now, in this scenario of high complexity at molecular level and high heterogeneity, what we need to do is to represent such complexity, such heterogeneity, if we want to improve the approach of personalized medicine in cancer. And of course, we can try to identify the different molecular alterations that are present in colon, lung, or other tumor types by using the methods that we are usually running in our labs that are used also for clinical diagnostics, such as sequencing, real-time PCR, FISH, or immunohistochemistry. However, the time, the cost, and the amount of tissue that are required for this approach are not compatible with the clinical research, in particular in some settings. For example, for advanced non-small cell lung carcinoma, in most cases we have available very little amount of tissue, and it will not be possible to perform all the analyses that are listed in this slide in the majority of the cases. This is why the cancer comprehensive centers are now trying to implement in their clinical research setting new platform, high-throughput platform, in particular genotyping platform and next-generation sequencing approach. Now, the main difference between genotyping and next-generation sequencing is that genotyping allows only to identify the point mutations of small indels and in particular only known mutations. In contrast, next-generation sequencing can identify known and new mutations in all the regions of the DNA that are amplified, so can provide much more information. Of course, next-generation sequencing can be also used for whole exome or whole genome sequencing, or can be used only for targeted sequencing. Well, what I believe is that now, for a long time, and really for a long time, the most used application of next-generation sequencing will be targeted sequencing that will allow multiplex hotspot mutation testing to assess at least the most common mutations, and more comprehensive molecular characterizations like whole exome or genome will be limited to those cases that are negative to the first level of screening, unusual response, so to a limited number of samples. Well, starting from these points, 8 European laboratories decided to collaborate in order to develop a dedicated panel to study hotspot mutations in colon and lung cancer. The idea was to generate a panel that was specific for colon and lung cancer. This is because most of the available panels contain a number of genes that are not relevant to these two tumor types, but also miss some of the genes that are relevant to these tumor types. In addition, we want to develop an assay that could work using a very small amount of input DNA. In particular, it was able to work by using DNA derived from formalin-fixed paraffin-embedded tissue. And finally, we want to generate a panel that could be adapted by other research labs, so that should to be accurate, affordable, and easy. And this is one of the reasons why we tried to multiplex as many samples as we could in the same chip. And the first set of data that I will show you, that in this case 5 samples have been multiplexed in a 316 chip. This is the list of genes that are analyzed by this panel, and this, as you can see, shows includes a number of genes that are involved in pathogenesis of both colon and lung cancer. With respect to the AmpliSeq cancer hotspot panel version 2, this panel includes 2 genes that are not included in the other panel, which are DDR2 and MEC1, and the number also of exons that are also relevant to these two cancer types, in particular KRAS exon 4, EGFR exon 12, and BRAF exon 11. So the study that we performed to validate this panel was organized in 3 phases. The first phase was aimed to assess reproducibility and accuracy of the panel, and in this phase the same 5 control samples were analyzed in the 7 different labs. The second phase aimed to assess concordance, and therefore 10 blind samples were assessed by each of 6 labs. And the third phase was aimed to evaluate analytical sensitivity of the panel, and therefore 6 labs were asked to analyze each 15 difficult samples, in particular samples with a very low percentage of tumor cells, so with a high level of tumor heterogeneity. The data were analyzed by using the ion reporter, and in particular a specific workflow has been elaborated, and a variome knowledge base specific for this panel has also been generated. Now these are the source phase 1, as I told you, 5 different samples, 2 xenografts, 1 lung carcinoma, and 2 achromatics control were analyzed by 7 different labs. Here is the list of the mutations that were expected in these samples, and all the 7 labs were able to identify these mutations. Of course, we are running a next generation sequencing panel, so we were able to identify also unknown mutations, and these are the list of 5 novel mutations, new variants that we identified in the D2 achromatics control. All these 5 variants were confirmed by Sanger sequencing, so confirming the specificity of the panel. In the second phase, each lab, actually 6 labs, received each 10 samples that were assessed by another lab of the consortium for the presence of the most common mutations. And again, all the labs were able to identify all the expected variants in these 60 blind samples. In particular, these are 10 samples in phase 2 were analyzed in my laboratory. These samples came from the University of Verona, and they have been characterized for KRAS, EGFR, and beta-catenin mutations, and we were able to identify all the expected KRAS, in particular 5 KRAS, 3 EGFR, and 2 beta-catenin mutations. Of course, we identified also other variants that we are now confirming with orthogonal techniques. The third phase was, I would say, the most difficult phase, in which every lab was asked to analyze 15 difficult samples, 5 colon carcinoma and 10 lung carcinoma samples. They were chosen for the low percentage of tumor cells or the poor quality of DNA. So we wanted really to assess whether the panel could work in limiting situation. And what we found basically that all the labs were able again to identify all the known mutations. There's only one mistake in this slide, because one of the EGFR mutation that was missed, but this mutation had a frequency of less than 2%, and what we assessed is that the sensitivity of our test is 2% for the hotspot mutation and 5% for mutations that are outside the hotspot. But what I want to show you are the results of my lab. These are the 10 lung carcinoma samples that were assessed in phase 3 in my lab. Many of these samples had a low percentage of tumor cells, 20 to 50%, and all the expected mutation, in particular 4 EGFR mutation, were identified. But what I want to underline is that if you use these type of panels, you can immediately have information on both intertumor and intratumor heterogeneity. In fact, by using the panel, we identified among these 10 samples 4 EGFR mutant, 1 KRAS mutant, and 1 BRAF mutant. So we could classify 6 out of 10 of these tumors in already known subgroups, defining the molecular-defined subgroups of lung carcinoma. However, we also obtain information on tumor interheterogeneity. For example, in the sample number 10, this sample was confirmed to carry a deletion of the EGF receptor in exon 19, which is associated with sensitivity to tyrosine kinase inhibitors. These samples also contained a mutation in the PIT kinase gene, which has been associated in preclinical and some clinical studies to potential resistance to anti-EGF receptor agents, and so this information could be important also for the response of the tumor to these agents. When you use these panels, sometimes you find some surprise. This is what happened for us when we analyzed the 5 colon carcinoma samples. One of the samples, the number 15, was found to carry a mutation in KRAS. That means was confirmed. We knew that this sample had a mutation in KRAS, but we also detected in this mutation, in this sample, 2 mutations in the EGF receptor, the T79M in exon 20 and the L858R in exon 21. So this was a big surprise for 2 reasons. First, for what we know, at least from lung carcinoma, usually KRAS and EGFR mutations are mutually exclusive. In addition, EGFR mutations are very rare in colorectal carcinoma, and so we were really surprised to find 2 EGFR mutations in these samples. I have to say that when we saw these results, we really thought that this could be an error of the panel, so it could be a false positive. However, my postdocs were able to confirm the presence of both the T79M and the L858R mutation in this case, so confirming again the absolute specificity of the test. Well, however, we were really happy about these results. However, when we analyzed the Amplicon coverage, what we found is that there is a small group of amplicons for which the coverage is quite low, and this might result in a reduced sensitivity of the test. And this is also on the right of this slide, there is a group of amplicons for which there is a very high coverage that is really unnecessary in this case, but there is a coverage that might overcome the capacity of the chip, and this might lead to problems, of course, in sequencing. So we took advantage of the fact that Life Technologies had available a new workflow for the design of primers, and we asked LifeTech to redesign the primers of our panel. And this new design was again assessed through different phases. We repeated phase 1 for reproducibility and accuracy, and also we went again to phase 3. But I want first to show you the coverage that we obtained with this new design of the panel. We have a more even coverage with an approximately 1000 coverage for each amplicon that ensures a very high sensitivity of the test. And another important point to underline that by using this new design, we are able to pull at least 8 different samples on a 316 chip. And looks like there is echo. Okay, I hope you can hear me. However, I can tell you that due to the capacity of the chip, most likely you can pull 10 and maybe up to 12 samples in the same chip. And this is again the validation that we did. This is the phase 1. 7 different formally fixed paraffin embedded tissues were assessed in 7 different labs, and all the expected variants were confirmed. And this is the phase 3. Again, the 5 labs reanalyzed the 15 difficult samples, and all the expected variants were identified in this phase. Now, to conclude the first part of my presentation, I think that the panel that we have developed meets most of the requirements of a test for mutational screening. First, this panel has shown a robust performance using limited amounts of formalin fixed paraffin embedded material, only 10 nanograms of DNA, and showed high accuracy and an adequate sensitivity for mutation with potential clinical relevance. As I told you, approximately 2% sensitivity for hotspot mutations and 5% sensitivity for mutations outside of hotspot regions. The results are available in few days. This is a flexible tool that can be easily implemented, and I'll tell you something about in the end of my presentation, and that more importantly allows to detect a huge number of mutations in a single pool. I mean, with this pool we can detect more than 500 known mutations in the 22 genes that I have listed to you. Now, I want to show you one application that we have recently made of this panel to our research, in particular to clinical research in colorectal carcinoma. As you might know, a number of studies have clearly demonstrated that mutations in KRAS, in particular these 7 mutations of codon 12 and codon 13 of exon 2 of KRAS, are associated with resistance to anti-EGF receptor monoclonal antibodies in metastatic colorectal carcinoma. Well, the principle is quite easy. When you try to block the EGF receptor with a monoclonal antibody, I mean, if KRAS is mutant, KRAS will be always activated, and therefore, if you block the EGF receptor in presence of a mutant KRAS, there will be no effect on the signal transduction pathways that are below RAS. And in other words, RAS is a kind of switch that connects the receptor to the intracellular circuits. Now, more recently, this study, published a few weeks ago in the New England Journal of Medicine, has demonstrated that other mutations in KRAS, in particular exon 3 and exon 4, as well mutation in NRAS, exon 2, exon 3, and exon 4, might also be associated to resistance to anti-EGF receptor monoclonal antibodies in metastatic colorectal carcinoma. I would say, however, that we expect that other molecular alteration might affect the response of tumors to anti-EGF receptor agents. Indeed, metastatic colorectal carcinoma have a complex molecular alterations, and many tumors carry different molecular alterations that we believe might have effect on the response to therapies as well as on the prognosis of the patients. For this reason, we decided to perform a wide molecular screening within the individuals that were enrolled in the CAPRI-GOIM trial. The CAPRI trial is a randomized trial whose principal investigator is Fortunato Cerdillo, who has presented this data a few weeks ago at the ESMO meeting in Amsterdam. While this trial enrolls individuals with a metastatic colorectal carcinoma that is wild type for mutations of KRAS in codons 12 and 13, as assessed by local pathology laboratories. These individuals receive as first-line treatment a combination of cetuximab, which is an anti-EGF receptor monoclonal antibody, plus a polychemotherapy FOLFIRI. At progression of the disease, the individuals or the subjects enrolled in study are randomized to receive either FOLFOX alone or cetuximab plus FOLFOX. The primary endpoint of the first and the second part of the study is the progression-free survival, and actually this is a strategy trial which is trying to identify those tumors that might benefit of treatment with cetuximab even after progression of a first line containing an anti-EGFR monoclonal antibody. Now, these are the main features of the subjects enrolled in this study, and these are compatible with what you really find in your clinical practice in the metastatic colorectal cancer setting. These are the outcome data of the study. We found a response rate of 56.4% and the median progression-free survival of 9.9 months. And this is really what you expect in a population of subjects with a metastatic colorectal cancer that are selected for being wild type for mutations of KRAS in exon 2 and are treated with the chemotherapy of cetuximab plus FOLFIRI. We had available 182 tissues from these individuals enrolled in this study, and we analyzed this tissue with the lung and colon cancer panel that I presented to you today. One first important point is that the analysis was successful in all these samples. And in these samples, we were able to identify 206 mutations distributed in 14 genes out of the 22 included in the panel. In particular, we found at least one mutation in 124 cases out of 182, so in the 68%, whereas almost 32% of the cases showed no mutation for any of the 22 genes that we analyzed. And these are the results showing the frequency of the mutation, at least for the most frequently mutant genes that we found in our subset of cases. Now, the frequency of mutations of this gene, P53, KRAS, PAT kinase, BRAF, and RAS, MET, FBXW7, are in line with what has been observed in other studies and what can be found in a number of databases. The only surprise was that we found 30 mutations in codon 12 or 13, so in exon 2, in 29 cases. And this was kind of a surprise because these tumors have been selected for being wild type at codon 12 and 13. How do we explain this? Well, first of all, it is possible that our technique is much more sensitive from the techniques that have been used by clinical diagnostic labs. In particular, in Italy, many labs are still using Sanger sequencing to detect mutation in the codon 12 and 13, and there is a huge variability in the sensitivity of Sanger sequencing between different labs. The other point is also that there are, at least for some cases, it's possible that to be analyzed a different specimen as compared with the specimen that has been analyzed to enroll the patients in the clinical trial. However, we also to acknowledge that it's possible that some of these difference might be due to errors that have been performed by local labs. Well, but what I want to really to underline you that by using our tool, we are able to better represent the complexity of these tumors. For example, we are used now to classify the colorectal carcinoma and metastatic colorectal carcinoma individuals in KRAS and RAS, BRAF, PAT kinase mutant, and so on. However, if you look, for example, at KRAS, 30 out of 45 cases that are KRAS mutant also carry different mutation in different genes, and there are many samples that carry 2 or more mutations with up to a maximum of 5 mutations. So what we really believe is that each one of these cases, each one of these different molecular profiles might be associated with a different outcome and with a different prognosis, with a different response to therapy, and not only to biological therapy, maybe also to chemotherapy. And we believe that we need to go into this detail if we want to better select patients that might benefit of specific treatments. Well, with regard to the outcome, what we found is that basically this slide just shows that the response rate and the progression-free survival of the population that was analyzed for the 22 genes, 182 samples, is similar to the general population of 340 individuals. More importantly, when we separate those cases that have mutations in either KRAS or NRAS from the cases that do not have mutation in KRAS and NRAS, we can see significant difference. In particular, in the KRAS NRAS wild-type population, the response rate passes from 57% to 62%, and the progression-free survival improves from 9.8 months to 11.1 months. More importantly, in the KRAS and NRAS mutant population, we found a very low response rate, 46.6%, and lower median progression-free survival, 8.9 months. So we really were able to identify those tumors that are likely to be resistant to anti-VEGF receptor monoclonal antibodies. Indeed, the figures that we get in this population of KRAS and NRAS mutant with regard with the response rate or progression-free survival is what we expect if we use chemotherapy alone. So these tumors are really likely to be resistant to anti-VEGF receptor monoclonal antibody. Now, if we add to this population also the BRAF and the PI3 kinase mutant, the progression-free survival drops to 7.7 months. However, we don't know whether this is due to resistance of these tumors to the treatment with the anti-VEGF receptor monoclonal antibody, or rather might be related to a negative prognostic significance of these mutations. In particular, we know that individuals with BRAF mutations have very poor outcome, and also individuals with double mutation, the PI3 kinase in exon 9, 20, have a very poor outcome, and we had 2 patients with double mutation in our cohort of patients. So, the conclusions of this study are in 3 levels. The first is just a clinical conclusion, is that in this in a population selected for only KRAS exon 2 mutations, we obtained the same results that have been previously shown in the registered trial of cetuximab in metastatic colorectal carcinoma, the CRYSTAL trial. More importantly, we have clearly demonstrated that the molecular profiling of colon carcinoma by using the AmpliSeq colon and lung cancer panel is feasible on FFPE tumor tissue. In particular, we received these 182 samples from many different surgical pathologies, over 20, and that this samples were fixed and treated and handled in many different ways, but the system, our panel, showed to be highly robust, and in fact the analysis was successful in all the cases that we analyzed. More importantly, by using this approach, we can represent which is the molecular complexity of these tumors, and we believe that this might have important implication for understanding better what is the outcome of this patient and what is the efficacy of treatments in this particular setting. The last point that's more important is that we show that by using this panel we can select those tumors that are really resistant to anti-VEGF receptor monoclonal antibody because they either carry KRAS, NRAS, and maybe also BRAF or PI3 kinase mutations. Well, this is my last slide in which I will try to summarize what we have done, but also to give you some insights on the future. Well, first of all, we acknowledge that this panel is able to provide us information that are limited to somatic mutation. Now what we are doing is to try to improve the panel in order to get also information on the copy number variation. In addition, we are now trying to develop a new panel that is based always on the AmpliSeq technology to study fusion genes that, as you might know, are particularly important in the pathogenesis of lung carcinoma. About 5% of lung carcinoma have fusion genes, and there are some data suggesting at preclinical and clinical levels suggesting that the tumors carrying this molecular alteration might respond to specific agents. And what we believe is that having available these tools that can allow us to perform a complete molecular profiling of tumor, starting even from a very little amount of DNA, and hopefully in the next future starting from circulating free tumor DNA, or even from DNA that we can extract from circulating tumor cells, well, we believe that by this approach we can provide a complete molecular profiling of the tumor, and this might be useful really to realize personalized medicine in cancer patients. Now, I cannot finish my presentation without acknowledging, first of all the colleagues that have participated to the development of this panel, including also people from Ion Torrent team that really have been very collaborative and pushed us to get this result as soon as possible. I've also to acknowledge people from my laboratory, in particular on the right are my collaborators in the Laboratory of Pharmacogenomics in Crom in Avellino, who have been directly involved in these studies. And finally, I guess I have to show you this disclosure saying that the Ion Torrent products discussed in this presentation are for research use only, not for using diagnostic procedures. And this is actually what is according to Life Technologies rules, to United States rules. We know that in Europe the situation is slightly different. I finished with this, and I will give back the presenter to the presenter the control, if I manage.
Thank you, Dr. Normanno, for a very interesting presentation and for sharing the work of the consortium and of the work in your laboratory. At this point, I'd also like to remind those that are attending the presentation that if you'd like to receive more information on the Ion Torrent technology, the AmpliSeq panel technology, or specifically about the lung and colon cancer panel that Dr. Normanno spoke about, you can learn more about these in the Life Technologies booth here at the virtual bioconference live seminar series. Next, I'll go to Q&A. Our first question is from Chinapa. What is the lowest allele frequency that can be detected using your gene panel sequencing method? Can you detect down to 2 to 3% allele frequency?
Well, as I said, we have evidence that at least for the hotspot mutation, we can go down to 2%. For mutations that are outside that spot, I mean, we are feel confident that 5% is the limit of detection. Below that, I mean, I think that we will need some confirmatory studies. And I think that also LifeTech is trying to work in order to improve the sensitivity of the method, and we know it's very important if you want to study tumor heterogeneity.
Thank you, Dr. Normanno. I have a second question also from Chinapa. What filtering steps do you employ to reduce false positives due to chemical fixation of FFPE samples?
Okay. Me? Okay, I mean, I'm not really the person who can tell you about this. What I can tell you that we did a lot of work together with Life Technologies people in order to make an adequate filtering of the data, and that now is available on the web when you have access to Ion Reporter. This filter that has been developed in order to ensure a high specificity of the samples. However, Kisanti Ainali from LifeTech is really the person who worked on this, so I think that she's the only one who can really address your question.
Thank you very much. And at this point, we will go ahead and close the presentation, and we wish to thank everyone that participated today. Thank you. Welcome to the Bioconference Live Virtual Cancer Conference focused on research, discovery, and therapeutics. My name is Michaeline Bunting, and I will be your moderator for this first keynote presentation. On behalf of Life Technologies, I'd like to introduce Dr. Nicola Normanno. Dr. Nicola Normanno is from the Center for Oncology Research from the National Institute of Cancer from Naples, Italy. He will be speaking on building an ion AmpliSeq colon and lung cancer panel, consortium development of a tool for clinical research applications. During Dr. Normanno's talks, we'll be accepting Q&A, of which I will facilitate at the end of the session. At this point, I will turn the presentation over to Dr. Normanno.